See, there is a term codominance. Let me explain what is it. This we can understand by going through certain examples. But see, when Mendel did his experiments by making crosses between those plants which possessed some contrasting traits, then he found that one of the characters was expressed in the next generation, in the F1 generation, and the other one remained unexpressed. So the feature which appeared in the next generation was dominant one, and that one which remained hidden was considered as recessive one. So he gave the concept of dominance and recessiveness. But codominance is a different aspect in which actually both contrasting traits are expressed in the individual. So let us see how such type of inheritance pattern take place. So codominance is a type of inheritance in which two different alleles of a gene locus, suppose the two different alleles are capital A and A1. These are two alleles of a gene locus. They are fully expressed in the phenotype of a heterozygote. It means when both alleles A and A1 contribute equally to the appearance of a phenotype, then the phenomenon is known as codominance. And this we can understand in case of Yemen blood group. See, very less people may be aware with this type of blood group because it is not of pathological significance. Actually, it does not cause any immunological reaction at the time of blood transfusion. So this has not been given much significance. Otherwise, there is a specific locus on the fourth chromosome which harbors two alleles. Those alleles could be LM and LN. L stands for Landsteiner, who actually discovered this factor, and the alleles M and N, they actually determine the blood group of the individual. So there is a Mn blood group system in human, which is quite often considered to explain certain population genetical aspect. Actually, in our population, in human population, there are three different types of individuals. Individuals having M blood group, or certain individuals may have N blood group, or there could be persons with Mn blood group. So three types of persons are there. And the Mn blood group is decided by the presence of two different types of native antigens. Okay, such antigens are actually located on the external surface of RBC. So in this diagram, these three structures are RBC, red blood corpuscles, and on their surface a native antigen may be present, that is actually glycoprotein structure. So those who have blood group M, they will have a specific type of native antigen. Those with N blood group, they will have a different type of antigen present on the RBC surface. And Mn type people, they have both M as well as N antigen on the RBC surface. So in this way we can say that there are two alleles in the population, M and N, and because of these two alleles, three different types of genotypes will be possible. That is MM, N, and MN. Okay, three different types of persons are there with respect to their genotypes as well as phenotype. This is actually beauty of this analysis, that if you know the blood group of a person, then exactly at the same time you can know the genotype of that fellow also. That is, those who have M blood group, their genotype will be MM. Persons with N phenotype will have genotype NN, and Mn blood group types, they are actually heterozygotes. So you can say that when a person with blood group M gets married with another one having blood group N, then they will have all their children with blood group Mn. So it is a case of codominance in which both antigens are being expressed on the surface of RBC. Other example could be of ABO blood group system, with which we are aware very well. See, this ABO blood group system is actually decided in our population by the presence of three alleles, that is IA, IB and IO. I stands for isoagglutinogen. Okay, this is also a kind of antigen present on the surface of RBC. And the blood group deciding, ABO blood group deciding gene is located on the 9th chromosome. Okay, so see those individuals, and we know that IA allele is actually codominant to IB, means both are able to express them, whereas IA, IB both are dominant over IO. So just to explain that which are the two are codominant, equal to this symbol is used, and both are dominant over IO. So by writing this, we can understand their nature. And individuals who are of blood group A, they will have a specific type of actually oligosaccharide chain attached with the lipid. Okay, so this is, or you can say, such lollipop-like structures which are shown on the external surface of RBC are actually the deciding factors for blood group. If the oligosaccharide chain has actually N-acetyl-galactosamine, suppose this is a oligosaccharide chain in which one of the monosaccharide is N-acetyl-galactosamine, then the individual will be of blood group A. And if this unit is having galactose, then the blood group will be B. And those who are of AB blood group, they will have both types of antigens present on the surface. That is oligosaccharide chain with N-acetyl galactosamine as well as with galactose. So this is also one of the best examples of codominance, where both factors are actually being expressed on the RBC. That is why I have shown here that the phenotype AB, whose genotype will be IAIB, such individuals are actually codominant type. We can also consider a case of cattles, that is coat color inheritance in cattles. See, in cattles we find that cattles may have completely red color, pure red color of their coat, of their skin. Their genotype will be actually capital R, capital R. Suppose in the presence case, those who have genotype capital R on a specific gene locus, they will be looking red in color. The other one are white in color, and their genotype may be shown as WW. So they are white phenotype, means their skin coat color will be white. So if you cross a pure red with white one, you will find in next generation cattles having mixture of both color, that is red as well as white. And such individuals, heterozygous individuals, are actually roan. They are referred as roan, R-O-A-N. And genotypically they are RW. Okay, they possess both alleles. It means both alleles are expressed. If you carefully observe the coat color, you will find that red and white, both type of hairs are present on the skin of such cattle. So in this class, what is shown that two roan color individuals, one male, other female, if they are crossed with each other, then in next generation you will have 25% cattle with pure red phenotype, 25% with pure white, and 50% of them being heterozygous, they will be roan coat colored. So this is also one of the popular examples of codominance.